We begin. And today, I'm very pleased and greatly honored to introduce our keynote speaker, Dr. Bruce Stillman. Bruce, probably most of us has made the greatest scientific contribution to science, and his work greatly advanced our understanding of the mechanism, how the DNA in eukaryotic cell is replicated. And the Bruce's work has two major parts. The first part, and I think he used the SV40 system to study how the biochemical reaction occurred in the replication focus. The second part of his work is he studied the initiation of DNA replication in eukaryotes, and he discovered first DNA origin recognition protein called the ORC, origin recognition complex, and this finding actually basically open up the field of DNA replication initiation. And furthermore, and I really cannot overstate the importance of the cold spring harbor series of meetings. And he promoted the cold spring harbor meetings in the Long Island. And also it's very important, I think, he launched the cold spring harbor Asian meetings here in Suzhou. And I think his action in promoting the cold spring harbor meetings, promoting scientific exchange, will leave a great mark in science. Okay, now today we welcome Bruce give us a talk. And today, the title of his talk is the mechanism, regulation, and evolution of DNA replication origin specification in eukaryotic cells. Okay, let's welcome.
Right, well, thank you very much for the introduction, and thanks to the organizers for inviting me to present this this keynote talk. It's a great pleasure, and I'm really honored to be able to present some of the science that we've done, which really started when I first came to Cold Spring Harbor over 40 years ago now, but more recently, we started working on yeast in the mid-1980s. What I want to focus on is is a comparative evolution of this complex system, which is a fundamental biological system, and how studying replication in yeast can actually inform enormously about the process in eukaryotes in general, even to plants, animals, insects, and other organisms. It turns out that they're not all the same, and that's expected, but the comparison between them actually gives some significant insight into this process. So for those who haven't been there, this is Cold Spring Harbor Laboratory in all its glory, and my laboratory is buried in the trees in this somewhere in this region here, and I'm very fortunate to be able to work at Cold Spring Harbor and also lead the institution. So just to put this whole system in some perspective, the replication of DNA is a very complex system, and the reason is that these are eukaryotic epithelial cells. These are two epithelial cells. The nucleus is stained in blue, and within the nucleus is 2 meters of DNA if you stretch out all of the DNA in the 46 chromosomes. it is about 2 meters of DNA, 3 billion base pairs from each parent. The little blue marks here in this picture here are actually bacteria. So they're contaminating bacteria, presumably from somebody's mouth from this image. And it shows you the relative size of these large epithelial cells to bacteria. But if one looks at the cells, just in the proliferating cells in our body, if you just take the cells in the bone marrow, there's about 500 million new blood cells born every minute of your lifetime. And if the DNA was extracted from these, it would stretch 1 million kilometers. And that's a long, long way. And to show you how long it is, it's 25 times around the equator of the Earth. So every minute in your bone marrow, you're producing about 500 million cells, mostly red blood cells, but a large number of white blood cells as well. And this produces a massive amount of DNA. And of course, that DNA is divided into 46 chromosomes, so that the chromosomes are long enough that they have to initiate from many, many origins of replication. Otherwise, the process of replication would never complete in a cell cycle if there was only one origin per chromosome. So this has to be a very coordinated event, and I'll talk about that process. Now, in the mid-1980s, we started studying DNA replication in eukaryotes in parallel with my studies on SV40 DNA replication. That turned out to be a very good comparison. Of course, chromosomal replication is more complex than replication of a small circular 5,000 base pair circular DNA in mammalian cells. But we chose yeast for two reasons. One is that there was genetics available, but also there had been some work like this important paper here from really leaders and pioneers in the field of replication that did electron microscopy to show replication bubbles here, here. And these replication bubbles you can see are different sizes. And this emphasizes that there is temporal regulation of DNA replication. Some origins fire earlier than others. And this is a programmed system in all eukaryotic cells that actually corresponds, as we heard from Nancy Kleckner's talk, corresponds to the organization of the nucleus. Now, these replication forks, they start at origins of DNA replication, and these are marked, hypothetical ones are marked in red. And the ones that are here, like this one, have fired early. This one a little bit earlier than this one here. Some of them haven't fired yet and will fire late in the cell cycle, but some of them are destined never to fire. They're called dormant origins, and they're a backup system. If there is damage to the genome, they can fire, if necessary, in late S phase to complete replication before one enters to mitosis. And at all of these red arrows, there's a pre-replicative complex that's formed only in the G1 phase of the cell cycle across the entire genome. So in yeast, there are about 4 to 450 of these origins. And in mammalian cells, there's one of about every 100 kb. So there's about 40,000 of those in the, or 50,000 of those in the genome. So this is the process of replication and assembly of the pre-replicative complex shown in the middle here, and the initiation of DNA replication. In 2009, John Diffley's laboratory and Christian Speck and my laboratory, Christian Speck's lab and my laboratory, reconstituted the pre-RC assembly with purified proteins, which is ORC, CDC6, CTD1, and MCMs on origin DNA, and showed that this was inhibited by the cyclin-dependent kinases, which is the reason why this step, this complex step, which you'll see complex, only can form in G1 phase. Paradoxically, the activation of this complex here, and particularly the MCMs, which are in head-to-head juxtaposition, are activated by recruiting a whole bunch of proteins, including CDC45 and GINS, which forms the active helicase. And this requires the cyclin-dependent kinase and also the CDC7 DBF4 kinase. And so not only do CDKs inhibit DNA replication, but they also activate DNA replication. Hence, one has to progress through the cell cycle to go through this whole process. Now we know, thanks to work in a lot of laboratories, but particularly John Diffley's laboratory, that we can reconstitute DNA replication with entirely purified proteins. And in fact, not only replicate the DNA, but assemble chromatin on the replicating DNA with these proteins shown here. The red ones are proteins that we had identified using SV40 DNA replication, but the others had been identified either from yeast genetics or from studying replication in human or Xenopus egg extracts. And but nonetheless, all of these proteins were known, and John's lab was the first to put all of these together to show that they are necessary and sufficient for the complete replication of a DNA, and in fact, from origins of replication. So I'll have more to say about the initiation of replication. So we have been over the years studying the structural and biochemical mechanism of assembly of pre-replicative complexes, which I consider the most regulated process in the initiation of replication, but actually one of the most regulated processes in the cell. And back in 2005, when Christian Speck, shown here, who's an extremely talented biochemist, was a postdoc in my laboratory, we started in 2005 in collaboration with Huiling Li, who was then at Brookhaven National Laboratory, just down the road from Cold Spring Harbor Laboratory, and is now at the Van Andel Institute. We started collaborating, and this in 2005, the resolution of the ORC complex that we isolated in those days by Cryo-EM. Of course, there was a revolution in Cryo-EM for which there was a Nobel Prize in 2017. And this has now led to Cryo-EM studies where you get atomic resolution or near atomic resolution of these structures. And so throughout the years, we've published a lot of work on yeast, but also in collaboration with my colleague at Cold Spring Harbor, Limor Joshua Tor, and her laboratory, published a number of structures on the human replication complex as well. So this is the path of pre-RC assembly that is known. ORC first binds to the DNA, as we showed, Steve Bell and I showed back in 1992, and recruits CDC6 that was actually first shown by Chung Liang that binds to ORC. And we showed in the early days of Cryo-EM that this DNA is bent in the ORC-CDC6 complex. There are now, as you'll see in a minute, higher resolution structures of this, which actually inform how it is bent. Then a first MCM-CTD1 is recruited onto the origin DNA. And then this process here occurs in the absence of ATP hydrolysis. It requires ATP, but no ATP hydrolysis. And then the OCCM, as we call it, the ORC-CDC6-CTD1-MCM complex, ATPases are activated in this complex, both the CDC6-ORC1 ATPase and the MCM ATPase, to form this structure where ORC moves over to the other side of the MCM, as shown by John Diffley's laboratory, to form this structure, and then recruit a second MCM chaperoned by CTD1 and CDC6 onto this to form head-to-head hexamer subunits. Now, the Cryo-EM of these, as I said, we started doing this in 2005. We first did the structure of ORC and CDC6, then the OCCM, and this was the first high-resolution structure, and it has been a gold mine, as you'll see, of information. And then a number of laboratories, including ours, have done the structures. Christian and Huiling and my lab have done the structures of this double hexamer. And the structure of this complex has only been done at this resolution, but all of the others have been done now at very high resolution, particularly ORC on DNA, by the Gao and Tai laboratories in China. Now, what I want to do is to show you some of these structures and show you how we've been able to tease apart the details of the mechanism of how this works and how it's made important contributions to understanding the evolution of origin specification in eukaryotes. So this is the ORC-CDC6 complexes. We realized very early on that they all contained AAA plus domains, which are ATPases, except for ORC6, which contains TF2B-like domains. They also contain winged helix domains, and you'll see the importance of these. And the AAA plus domains have a Rec-A fold, shown here, consisting of a Rec-A fold and a lid, which is the AAA plus domains, and a winged helix domain. There are sequences on both the the C terminus and the amino terminus, the C terminus and amino terminus of these that it's not shown here. And so these are the proteins that actually we got a crystal structure and a Cryo-EM structure with Lamore Joshua Tor of the human ORC complex and human ORC with CDC6, but also a structure that's now actually not in revision, but is now being accepted in Nature Communications from the yeast structure. And this is that structure. This is ORC and CDC6. And also in these proteins, the red parts here are intrinsically disordered regions of the proteins, which have an incredibly important role in the biology of pre-RC assembly, particularly in human cells, and I'll mention that at the end. So this is the ORC-CDC6 structure. The AAA plus domains of the 6 subunits, the 5 ORC subunits plus CDC6, surround the DNA in a perfect circle, whereas the the winged helix domains form a spiral around the double helix, shown here. So here's the spiral around the double helix, and reach out away from the A element of the origin into the B elements where the MCMs are going to be loaded. And the purpose of these winged helices reaching out along the DNA, it provides a platform for loading for the MCM proteins. And so this is the structure of the yeast ORC-CDC6 at 3.3 angstroms. This is now just been accepted in Nature Communications. And I should point out that Schmidt and Bleichert had published a similar structure from Drosophila, also showing DNA bending, but also the Li and Gao, the Gao and Tai laboratories did a structure of ORC alone on DNA, and ORC alone also bends this DNA somewhat. So this is the OCCM. This is the structure that we obtained in 2017. It's the structure of ORC and CDC6 on the top with the origin DNA shown here. And then the MCM, a single MCM complex with CTD1 shown in blue. This is formed in the absence of ATP hydrolysis, but with ATP. There's ATP in this structure. And if I show this movie, you can see that the DNA goes down through the channel of the ORC-CDC6, through the middle of it, and down through the channel of the MCMs between the yellow and the green, which is the MCM 5 and 2 subunits. And I'll talk about that. So you can see that the DNA, double helix of origin DNA, goes right through the middle of this channel. And so the MCMs and the ORC-CDC6 are topologically linked to the double helix. The double helix is not yet unwound, but it is kinked inside this structure and in preparation for its unwinding. So Christian's laboratory has been studying the biochemistry of this, and we have made some mutants, and the mutants have been very informative because they've been able to trap OCCMs, ORC-CDC6, CTD1 MCM complexes in various states of a semi-attached, a pre-insertion, and a mutant OCCM that's different from the wild type OCCM. And these have been informative about the mechanism and the assembly of the complex. The other thing that's really amazing about the Cryo-EM is that you can trap an apparent biochemically pure particle, but then one views it in the Cryo-EM, all of the individual particles, and separates them by computational separation. One can separate them into different structures that actually represent different states. And we've been able to do this even with the human orc with Lemore Joshua Tor, and trap them in different biochemical states of assembly of this complex. This is just one snapshot of the semi-attached OCCM, and the only, even though all of the proteins, the MCMs, the CTD1, ORC and CDC6 are present, the only parts that we could see in this Cryo-EM structure are this here shown on the left-hand side. The only parts of the MCMs that are visible are the two winged helices of MCM3 and MCM7, which also have AAA+ and winged helix domains. And this shows the interaction of the ORC2, 5, and ORC6, and you'll see CDC6 subunits of the ORC CDC6 complex interacting with the DNA. These are the winged helices that are reaching out along that DNA, origin DNA there. Now, this is the mechanism. I won't go through and show all of the Cryo-EM structures that we obtained for the reasons of time, but these are the structures that we obtained in this paper that was published in last year. And so, as I mentioned, the ORC CDC6 structure shows the DNA is bent. The two visible structures are the MCM3 and MCM7 winged helix domains, and this bit of the MCMs is floating around and is not in a stable position, hence it's not visible in the EM. The next step is that you get additional docking of additional MCM winged helices to the winged helix domains of ORC and CDC6. So the winged helices come together to form structures that bind these together. And first there's MCM3 and 7 binding to ORC6 and ORC4 and 1. And then the winged helix domains of ORC4 and of MCM4 and MCM6 come in and bind to the ORC CDC6 complex. Ultimately, then, there's a transition from this structure, as you'll see how that happens, of inserting the DNA between the channel of the MCM5 and 2. Prior to that, this channel is filled with these two MCM5 here and MCM4 here, winged helix domains, and you'll see that, ultimately forming this OCCM structure. And this is a computational model that was published in this paper based on all of the structures that were obtained. And so ORC and CDC6 are on the top and the MCM's on the bottom, and you can see the channel. And this is the first state where the DNA is starting to move into the channel, but the channel is filled with these winged helix domains that have to move out, the MCM winged helix domains that have to move out. So the DNA moves into the channel, and you can see that the interactions of this DNA with the various structures in the MCM, and then form this complex with CTD1 shown in blue still bound. The MCM4 winged helix domain shown in purple is moved out of this structure so that to accommodate the DNA. And you can see that when this moves out, it actually interacts with the double helix phosphates and stabilizes that structure. So this MCM4 winged helix has to profoundly move out, as does the MCM5 winged helix, move out of this channel to enable the DNA to get inserted. So the purpose of ORC and CDC6 bending the DNA is to allow the MCMs to come in and bind to the ORC CDC6, and then the DNA is inserted into the channel in an ATP dependent but ATP hydrolysis independent manner. So now the OCCM turned out to be unbelievably useful also, or valuable, for another reason. And this shows the assembly of the pre-RC, as I mentioned before. And the reason why we chose, John Diffley and I chose Saccharomyces cerevisiae back in 85, when we first started working on this, was because it was known, or it was suspected actually at that time, highly suspected, that it would have sequence-specific origins of replication. They were not known then until Bonnie Brewer's laboratory demonstrated that the ARS sequences were in fact origins. But Saccharomyces cerevisiae had these ARS sequences, and we proposed that these would be origins of replication. And York Marins in my laboratory showed the structure, the detailed structure of these origins. And at the same time, a number of people in my laboratory purified proteins that interacted with these origins, including John purified ABF1 and Steve Bell purified ORC. And this led, once we got ORC, we isolated CDC6 as a suppressor. That was Chun Liang's work, who's now at the University of Science and Technology. And that led to a lot of genetics to identify these pre-RC proteins. Now, it turns out that some yeast have gone through a whole genome duplication. That's these yeasts here. And that's what WDG is, GD is, it's whole genome duplication. So compared to, for instance, Clavomyces lactis, S. cerevisiae has duplicated genes. But K. lactis and all of these other yeasts have sequence-specific origins. These are all other budding yeasts down here. These are, this is Schizosaccharomyces pombe, a fission yeast, insects, humans representing all animals, and Arabidopsis representing all plants. All of these do not have sequence-specific origins. And this OCCM structure pointed to the reason why that is. So here are these conserved sequences. You can see that they're not all absolutely conserved, but we chose Saccharomyces to study. And the OCCM showed that there was an alpha helix of the ORC4 structure sitting in a major groove of the DNA. And there were other parts of this, including ORC2, interacting in a base pair specific way with DNA. So this was very informative. This was also confirmed with higher resolution structure of the ORCon DNA from the Gow and Tai laboratories. And this shows that alpha helix. Here's the alpha helix shown in pink. And these are the budding yeasts that I mentioned before that have that. And these are budding yeasts that do not have it here. This is the region where the alpha helix is missing, including in flies, in plants, and in human. And the reason why this was important is the comparative structures of ORC on DNA from Drosophila, from the Berger and Botchen laboratories, and also ORC on DNA from the collaboration we had with Lamore Joshua Tor, showed the following, that ORC4 structure is almost identical, not quite, but almost identical between Drosophila in gray, human in blue, and cerevisiae in pink, except for these two regions, this one here and this one here, which is the alpha helix that sits in that major groove, and as you'll see, forms base-specific interactions with the DNA. These ORC and ORCs from ORC4s from Drosophila and human, now at high resolution, have a small loop here, but no alpha helix. And this shows we did a massive parallel origin selection assay that was published earlier, and showed that we could isolate mutants in this alpha helix and do selection of origins of replication. And mutants, for instance, in this phenylalanine and the tyrosine, which interestingly, we converted those to the sequences in K. lactis. And then we showed that there was an AG to T transition, conversion of this dinucleotide into the TT dinucleotide, which is the dinucleotide found in K. lactis. So this was an interesting observation. And we did whole genome analysis of these using a checkpoint mutant that now shows all origins of replication firing in the presence of hydroxyurea. And you heard earlier in this meeting about the hydroxyurea inhibiting DNA replication. This is the wild type. So only the early origins fire in the presence of hydroxyurea. All of these late origins do not fire, except in the checkpoint mutant. And when we analyzed all of these mutants, we found that in fact, this mutant here never fires at an origin that has an AG dinucleotide in this position. And so this kind of validated with whole genome analysis, the plasmid selection assay. Then when we looked at the evolution of the ORC4 alpha helix, shown in this lane here, the ORC2 loop, which is also a base-specific interaction, and the sequence-specific origins, those species that have sequence-specific origins shown here, we found that this co-evolved with the SIR4. ORC interacts with SIR1 and SIR3 and SIR4 to silence gene expression in budding yeasts. And this is only present in the species that have defined origins. All other eukaryotes have RNAi-dependent gene silencing that you heard about from Shiv Grewal, that puts the histone mark on the CLA4 histone mark of H3K9 on silent DNA. And there is a transition here. Some species have lost, these species have lost the Argonaut and DICER. Some of these species, such as these, have remnants of DICER that is still functional, for Candida, for instance. But there's an interesting one, which is Euroia lipolytica, that does not have SIR4, and it doesn't have RNAi, it doesn't have sequence-specific origins. And a graduate student in my laboratory is studying the replication of Euroia lipolytica, because, as you'll see, we think this is going to be more like human cells than Saccharomyces. So now I want to turn to a comparison of the control of DNA replication in human versus yeast. And this is where things get really interesting and different, and is going to make textbook writing somewhat complicated. As you heard from Matt Logue earlier about the regulation of the cell cycle by cyclin-dependent kinases, this occurs in both human and in yeast cells. There's the CLINs and CLIB5 and CLIB6, and then the mitotic cyclins, not shown here, CLIB3 and others. They drive the cell cycle in budding yeast. In human cells, it is cyclin E. After cyclin D is activated at start, cyclin E, cyclin A, and not shown here, all of the cyclin Bs. Now, in yeast, ORC is stable throughout the cell cycle as a six subunit complex. In yeast, we showed that CDC6 is degraded at the G1 to S phase boundary, and it was shown in John's laboratory, John Diffley's laboratory, that is a ubiquitin-mediated process driven by cyclin-dependent kinases. Strikingly, and CDC6 and ORC1 are sequence-related. They're actually homologs, and in archaea, there's only one such protein, the ancient ORC1 CDC6 protein. But in mammalian cells, and particularly in human cells, it's a little bit different in different mammalian cells, but in human cells, ORC1 is degraded at the G1 to S phase boundary, just like CDC6 is in yeast. And CDC6 is actually degraded at the metaphase to anaphase transition by the anaphase promoting complex and by the complex in early G1. So this raised a paradox. If ORC and CDC6 are required for pre-RC assembly, they're only present in mitosis and in this period in the mid-G1 phase of the cell cycle when cyclin E is present. And so we began to look at this and ask, what is going on with pre-RC assembly in human cells? One of the things we found, and it was also demonstrated in David Gilbert's laboratory, is that ORC1 binds to mitotic chromosomes in early prophase as the nuclear envelope is breaking down. This is a GFP ORC1 showing it binding to mitotic chromosomes and is in fact inherited through metaphase and anaphase into telophase and into the G1 phase of the cell cycle. When it goes into the G1 phase, it forms these punctate spots in G1, and then this is a movie of this cell going through G1. And you see what happens to ORC1. It starts to form a pattern that then gradually disappears. And by the time that this movie is over, the cell is about to enter into S phase. So ORC1 is sequentially degraded throughout the G1 phase after it's loaded in mitosis. It binds the other ORC subunits in early G1, and we showed that it binds CDC6 in mid G1, just right about here. And that's dependent upon cyclin E CDK2. This is the pattern. And when I saw this pattern from my former graduate student, Nihan Kara, I pointed out to her that this exactly reminded me of the pattern of temporal regulation of DNA replication. That is, these patterns here look like late replicating patterns. And in fact, we proved that in this paper. She pulse-labeled cells with bromidioxyuridine, actually with a fluorescently labeled bromidioxyuridine after getting it into cells through transfection for a 15-minute pulse. So we pulse-labeled cells in S phase with BRDU, then took them through mitosis and did movies throughout G1 with YFP ORC1. And we looked at all of these movies and we asked, is there a pattern of replication, a temporal pattern of replication in S phase that overlaps with the temporal pattern of ORC1 in G1 phase? And in fact, that is the case. This just shows one such image, the BRDU pattern that was obtained during a 15-minute pulse in this S phase. And then later on in this G1 phase, this pattern, which is inherited, of course, it's part of the DNA, it's a nucleotide in DNA, overlaps substantially with the ORC1 pattern. You can see these patterns here, they're quite remarkable. Now, this is amazing because this is like standing on the side of a road and watching cars go by and then coming back the next day and see exactly the same cars going by if you do a movie. And so this overlap, and we showed many versions of this, this overlap shows that the patterning of ORC1 in G1 phase reflects the patterning, the temporal patterning of replication in S phase. And I want to remind you, and it came up during Nancy Kleckner's talk, that this is reminiscent of the structure of the TADs in the nucleus of cells. Now, the other thing we found in this paper that we just published last month is that when ORC and CDC6 get together, we had anticipated, based on the structures that were obtained, that the AAA plus proteins would be the principal drivers of interaction between ORC1 and CDC6, which bind to each other intimately. In fact, ORC1 has to bind to CDC6 to activate the CDC6 ATPase activity. But in fact, in a big surprise, we found that two short regions within the intrinsically disordered regions of ORC1 and CDC6 actually form a complex. And the AAA plus proteins cannot bind by themselves. The domains cannot bind by themselves. Furthermore, these IDRs from ORC1 form a liquid-liquid phase transition that drags CDC6 into it. And this is a phase transition that we can assemble in vitro. And that brings the AAA plus proteins together to activate the CDC6 ATPase. And this only occurs in G1 phase, which you'll see in a minute. This complex also exists as cells progress to the G1 to S phase boundary. But what happens now is that the very same intrinsically disordered region that binds to ORC1 in CDC6 now recruits cyclin A CDK2 to phosphorylate this protein and cause the destruction, the SCIP2 dependent destruction of ORC1 at the G1 to S phase boundary, or as it progresses through G1. So CDC6 and ORC are only present in these two times in the cell cycle. We showed in this paper that they cannot interact because cyclin A CDK2 phosphorylate them in mitosis and cause their destruction. There's no SCIP2 here, so it can't degrade ORC1. But then later on, we showed that ORC binds protein phosphatase 1 through the intrinsically disordered region of ORC1 to dephosphorylate ORC1, allowing CDC6 to bind with cyclin E to promote pre-RC assembly. And the question, and I'll finish this, is how are origins specified in human cells? If there's no sequence-specific binding motifs in the human ORC, how are origins specified? And there is a lot of literature now, including all of these proteins that we have discovered binds to ORC1 and other ORC subunits, including gene regulatory proteins. We published a paper showing that ORC1 is involved in repression, RB-dependent repression. These two proteins are not published, but we're about to write that up. Cyclin-dependent kinases and protein phosphatase 1 in the recent Molecular Cell paper. We showed many years ago that it binds to modified histones and also heterochromatin protein 1. It was also shown by others that it binds to histone H4K20. And then the binding of these methyltransferases and histone acetyltransferases that we have shown. These remarkably all bind in the intrinsically disordered region of ORC1. And so we think that this is evidence of epigenetic determination of origins of replication. And in fact, there's evidence for that in the literature. So this work was done, the work on yeast was done by Elaine Hu, who is originally an undergraduate from Xiamen University in China and is now a PhD student in my laboratory, along with Yijun Xu, who has been a long time in my laboratory. So they did the yeast work. The human work has been done by Manza Hassan and Kula Kabala, principally with help from Mike Tramontano and Meng Huang. And the work on Euroia is beginning with from Nargis Ali. And we've collaborated with these phenomenal collaborators. I mentioned Christian Speck, Huilin Li, and these two, Justin Kinney and his postdoc Amar Tarin, have helped us extensively with quantitative analysis of our genomic sequencing, and also Lamore Joshua Tor. And the funding is shown here, and I want to thank you very much.
Right, we have an online on-site question.
Yeah, any question from online attendee? Masa, you have a question? Masa?
His mic is off. His sound, your mic is off.
Yeah, okay. So yeah,
yeah.
Thank you very much for Bruce. Very impressive talk. One thing that I could understand is that the ORC, you said the BLDU pattern during the previous S phase somehow determines the ORC location during the next genome phase?
Well, actually, I think it's the other way around. I think that ORC binding to histone marks on the genome determines where initiation of replication will occur. And it's been shown that there's co-localization of ORC with histone marks, H4K20, for instance. We showed H3K9, and also H2AZ has been demonstrated by ChIP-seq analysis. And so I think it's the ORC binding to the chromatin that determines where initiation of replication is going to occur, which then determines the pattern of replication. And so I think it's the other way around, that even though we showed BRDU first and then ORC, I think it's ORC determining the temporal pattern of replication. Now, that determines where origins are, and then it's been demonstrated by a number of groups, but there's a very beautiful recent paper from Dave Gilbert and Wendy Bickmore and others about RIF1, PP1 regulating the timing of replication. And I want to point out that PP1 we showed in that molecular cell paper, that PP1 binds directly to ORC1, suggesting that there may be some temporal dephosphorylation of ORC1 as it moves through S phase.
So that the ORC is degraded during G1 phase, right?
No, well, gradually it comes off early replicating chromatin early in G1 after commitment to cell division. This is after the cyclin D step. And then it comes off gradually and ends up on the centromeric heterochromatin only, on telomeric and centromeric heterochromatin in very, very late G1, and then is degraded and is reassembled, resynthesized and transported to the nucleus upon nuclear envelope breakdown.
So do you suggest that the ORC binding during early G1 determines where the early firing origins are located?
Yes, well, ORC binding to the condensing mitotic chromosome. So ORC, as I showed, ORC1 is bound to the mitotic chromosomes in early prophase, as soon as the nuclear envelope breaks down. So ORC is bound to those chromosomes. We're actually trying to determine exactly how. And then it is inherited into the cell. Then it binds the other ORC subunits. Then it binds CDC6 and forms the pre-RC. Then it's degraded by the cyclin A CDK2 when it comes up in late, just about to go into S phase.
And those origins are fired early in the S phase. And
those origins, the early origins are fired early in S phase, and the late origins, which tend to be heterochromatic, are fired late in S phase. And so there's this temporal order that's regulated by RIF1, which binds to PP1, which also binds to ORC1. So I think that there's a temporal regulation set up by this system that is, which we're examining. This temporal regulation is set up by this system that includes, when you delete RIF1, as you know very well from when you delete RIF1, the temporal order is still the same, but it's just very, very shortened. So the temporal order is formed by some other structure, and we suggest that it's the histone modifications and binding to ORC1.
okay, thank you.
Good.
Okay, online question, Brenda.
Yes, we have another question from Brenda.
Brenda, yeah, your question.
Hi, good evening, Bruce. I was just sort of intrigued when you started talking about the whole genome duplication in fungi and other cells, and but the potential relationship between sequence-specific and non-sequence specific origins. Is there something that we can learn about the evolution of?
Yeah, so in the paper we noticed, so in that paper, I didn't have time at this talk to go into that, but
yeah.
What we noticed is that this there's a co-evolution of sequence-specific origins, the alpha helix in ORC4, SIR4 being present, and actually John Diffley and I showed many, many years ago that SIR4 is related to nuclear lamins in higher eukaryotes. And SIR4 binds to a protein called EST1, which is a nuclear envelope protein. And that also appears at the same time. So we think that there's a mechanism to silence genes, the mating type genes, and prevent recombination of rDNA and telomeric DNA, which is what the SIR proteins do. That evolved prior to whole genome duplication. So it's present in K. lactis. Actually, interestingly, K. lactis does not have SIR1 and SIR3, but ORC binds directly to SIR4. So that shows that there's even been plasticity since the evolution of this clade of yeasts. The vast majority of budding yeast, like all of the candidas and the pichias and all of these other budding yeasts, Euroia, which is a very, very, very distant yeast, all of those do not have sequence-specific origins. They don't have SIRs. They have RNAi remnants, except for Euroia. And then fission yeast, as you heard from Shiv, and Neurospora have the RNAi, so do all other eukaryotes. So there's been a loss of RNAi, and what we speculated in this paper was that you need to have, in a high density genome like Saccharomyces, you need to have origins placed in a sequence specific manner in intergenic regions, because to avoid the complexity of R loops that are formed by transcription hitting replication forks. And in fact, that is the case in Cerevisiae. All of the origins are in intergenic regions. The origins, the transcripts tend to go away from them, or there's some mechanism to prevent them going in, like in the rDNA FOC1 story. And interestingly, in Euroia, it has about the same number of genes. We just recently did a high genome sequence of that. It has about the same number of genes, but they're spaced out. It's a 21 kb genome, and it's spaced out over 6 chromosomes. There's a lot more room, and actually a lot more repeats in that genome than there is in Saccharomyces. So I think the loss of repeats has also evolved RNAi. You don't need RNAi if you lose the repeats, except you need telomeric repeats and rDNA repeats. And those are the reasons why you have SERS, to stop the recombination of getting rid of those sequences.
Yeah,
So
thanks.
that's kind
Any
of
more questions from online attendees?
Oh, yes. Yeah. Hi, Bruce. I'll help to read out the question from Ling Lo Ju. Great talk. So what's the ORC1's application on mitotic chromosomes?
Yes, well, very good question. We have not, or we or nor anybody else have actually shown how ORC1 binds to mitotic chromosomes. However, as I mentioned, ORC1 does bind H4 K20 dimethyl, H3 K9 trimethyl, and in fact binds histones, other histone marks that we've shown and published. So we think that ORC is binding to the chromatin, ORC1, and to DNA in a sequence independent manner, intrinsically disordered region. And then the protein recruits other ORC subunits only after telophase. So we're now trying to map different sites in ORC and delete them, but it's actually quite complicated, because as we just published in the Molecular Cell paper, there are amino acid sequences called short linear motifs in the intrinsically disordered region that are highly overlapping. They actually overlap by 6 amino acids in a 7 amino acid binding domain, where 2 proteins bind to this same sequence, but the sequence is actually, the sequence dependence is different. And so this is going to be quite complicated, and it's all got to do with the type of thing that Martin Logue talked about this, the complexity of cyclin CDKs binding to substrates and other proteins, protein phosphatases and histone transferases. Histone methyl and acetyltransferases.
We have a question from the on-site attendee. Okay, do you have a question? Yes.
Hi, Bruce. Very interesting talk. I want to I wonder that what's the influence of the chromatin modification in budding yeast that influence the origin firing and the initiation of replication.
So budding yeast, fortunately, to make it a lot easier, doesn't have as much histone modification. It does have them, but not as much. And the influence on initiation of replication is not really that profound. Actually, what is known, interestingly, is that the SER proteins bind not only to the silent mating type information. The SER proteins, one of which is SER2, which is a histone deacetylase. So it deacetylates an activating acetyl group on histones that are involved in transcription. So the SER2 deacetylates those so that the genes are not transcribed. And those related origins, and there are quite a number of them as shown by Catherine Fox and Michael Weinreich, that these related origins tend to be late replicating and are a different class of origins than the rest of the origins in the genome. But for the most part, the sequence specificity of ORC in Saccharomyces has overridden the histone marks effect.
Okay. And another question is, the other ORC subunits in human cells are sometimes quite stable during the cell cycle, like ORC2.
Yes.
So does it something related with origin, firing, and the ORC1?
Well, it may well be. We have shown actually, but not published, that CDC6 can form a complex between CDC6 and ORC2 through 5, for instance. But I want to point out that the ORC subunits have other functions in the cell. We showed many years ago that ORC1 plays a role in centrosome duplication, that ORC6 plays a role in even cytokinesis. ORC1 plays a role with SUVAR H3K9 in repression of transcription of the cyclin E gene. And ORC2 and ORC1 bind the PLK protein kinase that's also regulating centrosomes. So they play a lot of different roles. And we've also shown that ORC subunits are bound to centromeres in mitosis. And if you mutate those, in fact, we just published this in eLife late last year, if you mutate or get rid of ORC2, there is defects in mitosis and nuclear structure. So we're investigating that nuclear structure defect because that's a kind of an interesting observation with respect to the timing of replication as well.
Okay, thank you very much.
Hi, Bruce. So ORC, as you already mentioned, they have many roles beside the assembly of pre-RC. Do you think it will also participate in the origin melting?
No, actually, I don't know. The origin melting is something that is under investigation. We're not actually doing that. I know Christian Speck's lab is doing it, and I presume John's laboratory is doing it as well. Exactly the mechanism of melting. I think that's going to be principally driven by the MCM proteins and regulation of those by the cyclins. And also what we showed many years ago is there is a complex after pre-RC assembly. There's a complex which we called the pre-IC, the pre-initiation complex. That's a DDK and CDK dependent assembly of proteins that are now known like CC45 to bind to the MCMs. And that is required for this unwinding as well. And so I won't say any more about that, but I think there's people are working on that.
Yeah, so the second question is, budding yeast, even there's no histone modification, but the origins is still believed to cluster in a three-dimensional way.
Yes.
So yeah, could you discuss more about on the origin clustering in mammalian cells?
Well, some of that is driven by the SIRF proteins tethering them to the nuclear envelope through the SIRF4 mechanism and EST1. But I also think that there's going to be because replication timing is so correlated with the TADs, these large topological activation domains, which, as you heard from Nancy, are assembled by condensins and cohesins, that there's going to be an intimate relationship between ORC and those. Origins tend to be organized based on these nuclear structures. And so it is very clear from the work of David Gilbert and others that the timing of replication correlates with TADs. And I think that that's going to go down to the molecular level with some kind of control of pre-RCs being placed in certain positions in these loops. Now, it's been shown by a number of laboratories that the MCMs can move along the DNA, but that's on naked DNA. Whether it moves substantially on chromatin is not really clear to me yet. But I think that the cohesins will bring these things together to form these structures. The other thing that we showed in the molecular cell paper is that ORC forms a phase transition. And this phase transition can also bring these structures together. And I think that's going to be important for the foci that you see, particularly in mammalian cells. So thank you for the question.
Hi, Bruce.
I think we should.
My question is about the transcription and origin selection. Many clues show that the transcription may have remodeling the origin selection. I want to know the transcription have any influence on the acquired selection or other have other influence on the origin selection?
Yeah. So it depends upon which transcription you're talking about and which species you're talking about. So it's been known for a long, long time that deliberate transcription through an ARS will prevent its function as an origin. So in yeast, sequence-specific origins avoid transcription. There's mechanisms like in the rDNA to prevent transcription going into the origin. In mammalian cells and in Drosophila, first showed by David McAlpine and colleagues, that when you chip ORC, it tends to chip in euchromatin to transcribed regions and to transcription start sites. I think of the fact of chromatin organization, that chromosomes are organized into euchromatin and heterochromatin. The euchromatin is organized to get accessibility to the chromatin through remodeling complexes to not only to transcription factors, but also to replication structures. Now, it's been shown in C. elegans from Justus Whitehouse's laboratory that the patterning of transcription in early embryo development in C. elegans follows the patterning, the temporal patterning of replication. So that suggests, that one paper suggests that the patterning of replication, euchromatin versus heterochromatin, occurs before there's any zygotic transcription. And therefore, I like to say that in audiences where there's a lot of transcription people, that replication is more important than transcription. So that's
Thank you.
The conclusion I made.
Hi, Bruce. I know that the time we overrun a bit, but there's one question from Hisao. Yes, one last question from Hisao. Please go ahead.
Yes. Yeah. So about ORC1 again. So you suggested ORC1 binding may be determining the replication timing. In the next S phase. So what will happen if you overexpress ORC1? Would it affect timing in the next S phase? And what will happen if you stabilize ORC1 during G1 phase, let it bind for a longer time on the during G1 phase? Does it also affect the replication timing?
So good question. And we actually just published that in that molecular cell paper that came out last month. You cannot overexpress wild type ORC1 in human cells because ORC1 causes the destruction of ORC1. So ORC1 binds to itself and regulates its levels in the cell. So you can't overexpress wild type ORC1 because its levels are very intimately determined in the nucleus. You can't even underexpress it either. So, but then we made a mutant that can't get degraded exactly as you suggested. This mutant kills the cell, the protein's not degraded, the nuclei get very, very disordered, and the protein's not degraded, and the cells will die of apoptosis.
So OC need to get degraded during G1 phase. That's for the proper replication?
Yep.
So the OC need to get degraded.
Yes.
For survival. And is that for the S phase, proper process progression of the S phase?
Yes, I think ORC1, if it's not degraded, will inhibit pre-RC assembly. In fact, we showed that. And so this temporal degradation through G1, if you don't do it, you don't form pre-RCs efficiently. We showed that in that paper. And therefore the cell is really confused because ORC1 actually, we showed many years ago in a science paper that ORC1 inhibits cyclin E CDK2. And in inhibiting, and cyclin A CDK2. So if you overexpress ORC1, it's like a CDK inhibitor and you're stuck. And the cells will die. Anyway, I think we should go on because I want to.
Thank you very much, Bruce.
Okay.
All right, thank you very much.
Let's thank the again.